Hello everyone, welcome back to my channel. My name is LEE ZHI XUAN. This video is the free class video for STPM Biology Semester 2, Chapter 14, Infectious Disease, which is also the last chapter of this semester. So this video is specially designed for those who've taken STPM Biology Semester 2. So let us see, based on the Majlis Peperiksaan Malaysia MPM, the syllabus for this chapter is quite short actually because we only learn about the type of the disease in the syllabus. So the first topic, of course, you need to know what is infectious disease. You need to define the infectious disease. And then the next one is the new topics, which is you need to learn about the vaccine using smallpox as example. So this is only the new syllabus add on. In the old syllabus, we don't need to learn about vaccine. But for now, you need to learn about the vaccine. And there are 5 types of the disease, infectious disease, you need to learn in the syllabus. So the first one is dengue, cholera, tuberculosis, malaria, and hand, foot, and mouth disease, HFMD. So in old syllabus, only learn about 4 disease only, dengue, cholera, TB, and malaria only. But for new syllabus, MPM add on the new disease, which is the hand, foot, and mouth disease. So each of the disease, you need to learn about the causes, what caused this disease, development of the disease, when the vector enter to your body, bacterial, virus enter to your body, what is the development, and also the symptom, transmission, and their prevention.
In this lesson, I'll be using these figures to guide our explanation. If you've already purchased my notes, you'll find the full, detailed explanations written out there to follow along. If you'd like to get your own copy of these notes, feel free to contact me directly.
Okay, so first of all, you need to know the definition of the infection. Okay, what is infection? Because you know that infectious disease, the first term is infection, derived from the infection. So when we say infection, meaning that it is actually the process when there are any non-self substance, including microbes, parasite, or any pathogen enter into your body. When these 3 things enter into your body, infection starting to begin. So it can begin anywhere in the body. When see lah, where is the pathogen go into? With like COVID-19 virus, it enter to your lung, right? So your majority infection is occur at your lung. And if more severe, the infection may spread throughout your body. So normally infection can cause fever and other health problems depending where it occur. So infection in early, it can be very small symptom, but in late stage it can be very serious. It can cause death one if you do not do the cure treatment, if you do not receive the correct treatment. So this is the definition about the infection. So this one is most important. Infectious disease, very very important. You in exam past year already came up many times. Normally, if the question especially in essay, when they want to come out this chapter essay, normally the first question is ask you to define on what is mean by infectious disease. So when you want to define it, there are 3 keywords that you need to learn. First one is it is caused by pathogens. This is very important. You need to learn it and transmitted through a vector. And also it is transmitted through from an infected person to an uninfected person or healthy person. These 3 keywords you must including in the definition. Normally 3 marks. These 3 keywords is each keyword 1 mark. If you lost any keyword, then lose mark. So this 3 keyword is very important. Okay, so this is an example as I mentioned. So this is an example of the infectious disease and their pathogen and transmission pathway. So this one is the vector type. Is considered as vector? So including virus, bacteria, fungus, and also protozoa, protozoa. Yeah. So in our syllabus, we only learn about TB, right? Tuberculosis. So tuberculosis is transmitted through the bacteria, and it transmitted. The vector is bacteria, and the transmission is through the air. And also the cholera is also transmitted by the bacteria. The vector is called bacteria, and then it's transmitted by the contaminated water or food. And another one is malaria and dengue. So malaria is transmitted through the Plasmodium species, which is a protozoa, and dengue is transmitted through the virus. And they are through the virus, which is their vector, and the transmission is by the mosquito, like mosquito. And the another one is and mouth, foot, and mouth disease. That one is also transmitted through the virus, which the vector is through the virus. And the transmission is through the air. Okay, so this is just example. I just give you a clue. Later we will go through more detail one by one. Okay. Okay, first one. Okay, this is a new syllabus that MPM add on, which a vaccine. You need to learn the concept of the vaccine using smallpox as example. So normally this is the history of the vaccine. I just briefing you what is the how the vaccine is discovered uh early from the 16th century. So normally the word vaccine is originated by the Latin word which is vaccine, and normally this word is discovered when the scientists called Edward Jenner they did the experiment to prevent the smallpox in human during 1798. Ha. After that, vaccine applied to all biological preparation produced from the living organism that enhance the immunity against disease, either prevent or in some case treatment the disease. So normally you need to know that vaccine is to enhance your immunity. The most important thing is enhance immunity. It is not a medicine. It is not a drug or medicine. Vaccine is not considered as medicine. I'm not saying that I consider medicine. Vaccine is not considered as drug. Drug. Why? Because vaccine, the main purpose is to increase your immunity. Drug normally is to kill the pathogen. Normally, it has the certain purpose one. Either you want to kill the pathogen or you want to kill the infected cell or any lah, based on the disease lah. But the vaccine normally is prevention method, meaning that even though you never get the disease, but you still can take a vaccine. But drug cannot. Drug normally only you take a drug, normally you get the disease, right? You get that disease, you consume the drug. But vaccine is prevention method. You don't need even though you do not have get that disease before, but you still can take a vaccine for prevention method. Like COVID-19 lah. Even though you do not have COVID-19, but you still can receive vaccine, right? This is the purpose of the vaccine. Okay. Vaccine. Okay, so let us see smallpox. Okay. Smallpox actually is one of the I can say that is one of the oldest disease in the world lah, caused by at least 3,000 years already in human. So this is example lah. Example of this one picture is from Wikipedia. If you search Wikipedia, you will search this kind of picture. This is very famous picture. Why we say it is smallpox because you can see their skin. Normally the person that get the smallpox, you will see there is a thick fluid, bump-filled skin like this. So you need to know smallpox actually is the acute and contagious disease caused by the variola virus. So smallpox is caused by the virus. It is caused by the virus, and normally it's already causing millions of death in the world before it was eradicated. Meaning that for now, smallpox is already eradicated. Meaning that there are no smallpox anymore in the world. This is declared by World Health Organization in 1980. Meaning that this kind of disease is already clear. There are nobody in the world now will get the smallpox. But still you can see the news. Some of the labs still maintain store this virus lah, variola virus. But whatever, you just need to know that in the world now there are no smallpox anymore. So how can WHO declare that smallpox is eradicated? There are no smallpox anymore. One of the function is vaccine. Because the discovery of the vaccine, the first objective to discover vaccine actually is to prevent smallpox. The first disease to to generate to discover the vaccine is using smallpox. So that's why smallpox can be killed in the world lah. Okay, so normally you can see that this is how vaccine work. So normally vaccine containing the component derived from a pathogen actually. So actually is still containing pathogen, but this pathogen is harmless. Or greatly attenuated, meaning that they already remove the harmful gene of the virus. Now, if I use the example for the smallpox, meaning that they still use the vaccine still containing variola virus one. They still containing variola virus, but they already remove the harmful gene. All the harmful gene they already remove. Remove. They just maintain the structure of the virus only, so that the component of the disease causing organism or the vaccine component will trigger the immune respond. So that it's still containing the virus antigen similar. But this virus now in the vaccine is already harmless. They will not infect the cell one. In normal situation, virus like HIV virus, it will automatically infect the cell. But for now, this vaccine, the variola virus in the vaccine is harmless. They just stay at there only. They just stay at the blood only. After that, your immune system, your helper T cell will go to there. It will just like normal situation, just renda or see go. Oh, see this virus. They just bind to the receptor, engulf the virus and display the antigen, so it will trigger the response by adaptive immune system. Adaptive immune system is what I already explained by last chapter. Development of the immunity, helper T cell, your macrophage, engulf the virus. After that, it display the antigen with the MHC2 complex, and the helper T cell will be activated, and then it will trigger the activation of the cytotoxic T cell for cell-mediated immune response, and the B cell for humoral immune response. So these are so the component of the vaccine determine the types of the vaccine that will show in the next page. So in the world now, there are many types of the vaccine, including live attenuated vaccine, inactivated, subunit, toxide, conjugate DNA, and recombinant. There are many type of the vaccine, but I think in the syllabus you don't need to learn. Uh, each type lah. Each type of vaccine, what is their mechanism? Actually, you don't need to learn lah. So if using the example, smallpox actually is using live attenuated vaccine, which this is still the live vaccine, but is less pathogenic. Less pathogenic, meaning that this virus, the virus is still same. They still alive. They still can infect other cell one, but it is less pathogenic. Pathogenic, and so it can induction of long life response. But there are also the disadvantages because it is still live, meaning that it still can trigger the infection of the cell. So this is a first type lah, and then after that, many scientists still will discover many different type of the vaccine. And also there are another one is mRNA. mRNA which now using in COVID-19. Another type is mRNA vaccine which now using in COVID-19 lah. But whatever in your syllabus, I don't think you need to learn all the type of the vaccine because each type of the vaccine will have different type of the mechanism to trigger the adaptive immune response. Okay. This is very important. Harmless pathogen will not attack your cell, but mimic the key feature of the pathogen so that it can activate the adaptive immune response. So what I say lah, meaning that it just stay at the blood only, in the blood only, or in the organ anywhere only, but it will not infect your cell. So that but it still mimic the key feature of the pathogen, like antigen on the surface, the glycoprotein on the surface of the virus. So that your immune cell, macrophage, and your helper T cell still can recognize this as non-cell, and then once they recognize it, meaning that your adaptive immune response will have memory, right? You will still remember they will proliferate into memory cell. So when you expose to the real virus, meaning that you already received the vaccine for smallpox virus. Okay, so now your body already have the memory to detect this kind of virus. This kind of they already have memory helper T cell, memory B cell, and memory cytotoxicity cell. So when you actually you in real time, in next time, you expose to the real smallpox virus, variola virus, and it is very harmful. It can infect the cell. When you receive it, be infected, then your immune system will be quickly respond to that virus. It will don't need to wait anymore because you need to know adaptive immune response is require very long time to activate it. Normally it takes about 70 hours, even up to 100 hours. Meaning that 72 hours already 3 days, 100 hours already 4 days, 5 days. You imagine if within this 5 days, this virus already can infect your many healthy cells already. But due to you have the memory inside your body, you can eliminate this infection, this virus rapidly before the pathogen infect to the body cell. So this is how vaccine work. Similar like COVID-19. When you receive the vaccine, you need vaccine, not say that you will never get COVID-19 any in the future. No, no such thing. This vaccine just enhance your immune system only. It just provide a memory to your immune. Okay, bagi tahu macrophage helper T cell ko awak ini benda baik, benda tak baik tau. Benda tak baik. Ini musuh tau. Musuh tau. So you already remember that. So after that, when the real musuh, the real virus coming into your body, you still can you will eliminate the virus rapidly. You can be activated more faster. So the effectiveness and the duration of the effect of the vaccine is based on the nature of the vaccine constitute and on the manner in which they are processed by the immune system. Okay, so this is the effect of the vaccine. Effect of the vaccine. So normally there are two factors that contribute the ability of vaccine to control or eliminate the disease. So the first one is effectiveness, and the second one is efficacy. Effectiveness of the vaccine, and then the second one is the vaccination coverage. So you can see here, this is a normal group, and this is the vaccinator group. Vaccinator group. Good. So vaccine efficacy is calculated in clinical trial by comparing disease rate between two groups, those who receive a placebo and those who receive the vaccine. So the vaccine efficacy typically measure the prevention of the disease in the clinical trial, and then the vaccine effectiveness normally is to assess the ability of the vaccine to prevent a specific outcome in real situation. So efficacy normally is performed during the clinical trial. Before you need to go through vaccine marketing in vaccine, you need to do a trial. So efficacy actually is normally done in the trial lah, human trial. And then effectiveness is that how do you determine that this vaccine can prevent the specific outcome in real situation, meaning that vaccine is already in the market now. Market now. So, vaccine may fail to induce immunity in a few individuals, but most effective vaccine induce a protective immune response in 95% of the individual. So, meaning that when your coverage of the vaccination rate is higher, more higher, meaning that this disease the population to get the disease is lower. So, you will get the herd immunity. Which is a form of the protection from an infection disease that occur when a sufficiently large proportion of the population get the vaccine. So you can see these three situations. The blue color one, the blue people is the healthy person, but they do not get the vaccine. So this is a normal situation, which if all are normal, but when there are some people get the disease like COVID-19, in COVID-19, because no vaccine as well, so all of majority of your population will get the disease, will get the COVID-19 very fast. This is very fast one. But when some of the people already get the vaccine, you can see vaccine, but the vaccination rate is very low. Only some people, only 4-5% get the vaccine. It is very low. The disease still can be spread. Still can be spread. But when the vaccination rate, many people already get the vaccine, even though there are some people already get COVID-19, but you still can protect the person that are not vaccinated. which we call as herd immunity, meaning that when more people get the vaccine, the number of susceptible hosts will be reduced. So the immune individual, meaning that the vaccinated individual, will act as the barrier in the chain of the transmission, make the vector, your pathogen, is very hard to transmit along the population. It like a barrier lah. So for now, if the vaccination rate is very high, using example as COVID-19, even though there are patients got COVID-19, but COVID-19, the virus now is very difficult spread to the unvaccinated person. So this is what we call as herd immunity. So if a high level of the vaccination rate is covered by a effective vaccine, well, disease of transmission will be affected, will be interrupted. Meaning that the disease want to transmit is very difficult. So that's why smallpox is become eradicated lah. Because everyone in the world now is receiving smallpox vaccine. At that time lah, especially in 1980, at that time. Before 1980, because if not mistaken, smallpox vaccine is discovered at here. Here. 1796, and then after early 17th century, many countries start to mandatory the smallpox vaccination. Starting lah, so that after the 100 year, you can see 100 year in 1980, WHO already declared that smallpox virus is already be removed in the world now. So that's why lah, vaccine, and this is caused by herd immunity. Similar like COVID-19. Why we say that when you get the vaccine is very important? Because you are not just protect you only, you also protect the whole population in the world. If only the certain people get the COVID-19 vaccine only, is nothing. You only protect yourself, but you cannot protect other people. So normally this one is to protect the person that cannot receive the vaccine. Normally it's very elderly person, or they have the critical illness like cancer, HIV patient, diabetes patient, all that patient, maybe they cannot receive the vaccine. So how they can prevent it from this disease? So herd immunity play the role. So that's why vaccine is very important. If you have vaccine be discovered that can treatment for the disease, then go ahead. It is no such thing that you need to wait or anything lah. But this based on the scientific study lah from the herd immunity. Okay, so that's all about the vaccine. Now we will go through the first infectious disease, which is dengue. Okay. I think dengue everyone know, right? I think because this is the one of the Malaysia famous disease in Malaysia. In Malaysia, because it's an acute arthropod-borne viral infection. Normally is the in the tropical and subtropical region lah. So as I mentioned, you need to know the causes of the dengue. So the infectious agent is virus. Infectious agent is virus. So dengue virus. So there are 4 types of the dengue virus. This DENV 1, 2, 3, 4, just like that. DENV 1, 2, 3, 4. And the scientific name for the virus is flavivirus. This is scientific name. Flavivirus. So if you want to write the scientific name of the scientific name, you can see in computer I italic the word, right? Italic the word, and this is the genus name, genus species name. So if you see the vector, the mosquito, right? It is transmitted through mosquito, right? And we know that the mosquito that cause disease is Aedes. So Aedes is actually the genus name of the virus, of the mosquito. Sorry. So there are 3 major species of the mosquito that spread the dengue. So including Aedes aegypti, Albopictus, and also Polynesians. So in exam, if the question asks you to write a vector, please write species name. If you write only genus name, you need to see the answer scheme lah. Some year answer scheme accept, but some year not accept. So it will be safe if you write full. So if you want to write full, the most important thing is you need to, the genus name, this first letter A, you need to capitalize. After that, all is small letter. And then you need to underline. And then the underline is not directly underline. You need to underline word by word. meaning that underline here and underline here. You cannot directly underline. If you directly underline it is wrong. It is wrong. You need to separate the line. At least one line, adjective, one line. This is very important. This is how you write scientific name in your exam. Because in computer you use italic. But for in exam you don't need to write like italic one, cannot. So in exam is you need to capitalize the genus name, which A must capitalize, capital word, and then other word all are small letter, and then you need to underline it. At least one line, adjective one line, similar to here. At least one line, epophytus one line. And then for scientific name of the virus, because no species, you just write flavivirus, capital F, and then underline it. That's all. So that's all. So this is a diagram for the Aedes mosquito, right? And then only female Aedes mosquito can spread the dengue. This is also one of the important. In exam, you need to write female. If you not write female, also wrong. So female important too. So this is diagram of mosquito lah, and then this is a virus lah. So it is also the envelope virus that containing RNA virus as well lah, as well. So this is a transmission lah. So how it's transmitted? So the vector is recalled as mosquito lah. Here, female mosquito, very important. So how they transmit normally is the female Aedes mosquito will take the blood from the infected person that containing the dengue virus, DNV, and then after that go through the another healthy person. Not only humans, some of them maybe can take from the animal as well, but we are focusing on human only lah. You don't need to like animal lah. Yeah, but in real world, of course, they can also infect animal as well. Okay. So this is a simple chart. I simplified the chart how the dengue virus transmitted. So normally, first of all, is when the Aedes mosquito, female Aedes mosquito, start to bite the infected person. Because female mosquito Aedes itself, it does not containing virus one. How this virus can be have present in their body only when they bite the infected person. Once that the Aedes virus starting to activate their own immune system, so that the virus can stay at their body. Normally is the gut of the mosquito lah. Meaning that the virus will not be killed by the Aedes itself. So the virus still can maintain at the mosquito body. After that, the virus will distribute throughout from the midguts of the body of midguts until to the blood of the mosquito, which we call as hemocell lah. Meaning that the virus starting to distribute throughout the mosquito body. So finally, it will go to the salivary gland cell, which is the extrinsic incubation period. The virus starting to replicate and mature to ready to transmit. So meaning that in the salivary gland cell, your dengue virus starting to reproduce large amount and be ready to be transmitted. So when this mosquito they containing dengue virus infect to the uninfected person, which is a healthy person, the dengue virus will directly enter into the host cell, which is a host body, this person. So the dengue virus starting to attach to the host cell. So the target cell lah. Later we will see the target cell. What is the dengue virus target cell? Like the HIV virus, the target cell is helper T cell, right? For dengue virus, the target cell, the target cell normally is white blood cell and platelet. White blood cell, platelet. So once they already bind to the white blood cell and the platelet, it was starting to reproduce inside the white blood cell and enter the cell by receptive mediated endocytosis, and then it will reproduce in the white blood cell and produce more and more dengue virus. And then this dengue virus will infect other type of the white blood cell as well. So this is an overall transmission process in the dengue virus. And the process is recycled. And then after that, when there are another mosquito bite this infected person again, then it will transmit again. So this process is cycle one. So yeah. So after when your virus enter into your body, what is happening? So as I mentioned, it will bind to the host cell. So the host cell for dengue virus normally is white blood cell. White blood cell. So that it will multiply at there, reproduce at there to produce more and more dengue virus and infect more and more white blood cell. White blood cell. So normally it will causing your lymph node and your lymph node and spleen become swollen lah. Swollen lah. Swollen lah. So development of dengue normally they have primary infection and secondary infection. If you got heard about that, when the person who get the dengue before, when they get the second time dengue, it will become more worse, more severe situation. Not like the other type of the disease, like one of the disease is chicken pox. If you see the chicken pox, if the person already got the chicken pox before, you will never get the chicken pox, right? Because your body already have immune system to that chicken pox virus. Chicken pox virus. So even though you will get the chicken pox virus enter into your body, you still can still your immune system will rapidly occur. But why this mechanism, the memory mechanism, does not apply to dengue? They have the reason why. So that's why they say primary infection and secondary infection. So normally, your primary infection is first time when you infected with dengue. So when you first time infected with dengue, just like normal situation only, like chickenpox one. They infected to your red blood cell and then reproduce all like that only. But for secondary infection, meaning that you already get the dengue before, already get first time or second time before, but you infected again. So this one is recalled as secondary infection. So let us see what is the cause the infection of the dengue. So this is a diagram that I try to simplify it. So when the person is infected with the dengue virus, we see the primary first. Secondary, you don't need to see first. We see the primary first. Okay, after your you first time get the dengue virus, your dengue virus infect the white blood cell. So imagine this cell is white blood cell. This cell is white blood cell. It will starting enter through the endocytosis process, cell-mediated endocytosis process. After that, it will replicate in the, it will release the viral genome, RNA virus, and then it will RNA virus, and then it will starting to replicate in the using your cell enzyme, including transcription and translation, and produce many viruses. So your helper T cell will recognize the dengue virus, and activate the adaptive immune response. So when at the same time also, your adaptive immune response still functioning. In the blood, helper T cells still can activate the adaptive immune response as well. So after the infection, your body already remember this infection forever due to the presence of the memory cell. So this is a first time infection. However, in the dengue infection, you need to know dengue have 4 type of virus. DNV 1, 2, 3, 4. 1, 2, 3, 4, right? So imagine if this person now is infected by dengue first virus, DNV1. After that, they're infected with DNV2. If they're infected by he or she infected by DNV2, it will cause secondary infection. And which although it is still a dengue virus, but the structure is different already. The structure is different. Okay, so if that person will have worse infection than the first time, which is what we say as it will cause dengue hemorrhagic fever, DHF. Dengue hemorrhagic fever. So if you cause by the first infection, it's just classical dengue fever only. But if second infection, you will cause dengue hemorrhagic fever. So when you want to write about the development of the dengue hemorrhagic fever, this is the key term. Caused by second infection by different dengue virus. If same dengue virus, DNV1, still same. You just say classical dengue fever only. You will not get this. But if you got the second infection and the dengue virus are different, then you will got this. So why this happened? Because of the antigen antibody enhancement ADE process. ADE process here. Antigen enhanced infection dependent process. Right. So these AD infection occur when the pre-existing antibody, you can see right, this here got antibody for the primary infection because they already have memory. Memory. So actually the shape is similar. The difference is the virus between the MV1234. Normally is the glycoprotein, the structure, the sequence, the genomic sequence. The genomic sequence are different, but normally the shape are similar. So your antibody still can bind to that virus. Okay, in general situation, if your antibody bind to this virus, you will do the antigen antibody reaction like agglutination, neutralization, precipitation, all that. But now all these process cannot be done. Now they still done this process. Agglutination, precipitation finally is engulfed by white blood cell as well, right? Engulfed by white blood cell. So your white blood cell will automatically engulf this virus automatically, because the dengue virus now is already be clumped together. They already be trapped by the antibody. So they need to remove, your body need to remove this virus through the phagocytosis by your white blood cell, including macrophage, neutrophil, basophil, all that. So your white blood cell will go to there and then engulf this bacteria. So when they engulf this bacteria, it will bind to the any infection will occur when the pre-existing antibody from the primary infection will bind to the dengue virus with different stereotype, resulting the antibody cannot neutralize that virus. This make them bind toward the FC receptor of the host cell. Host cell. So what I say the FC receptor is the host cell, like this is the FC receptor, so that it will enter into the white blood cell, and then the further infection mechanism is same as the capsid receptor in the infection mechanism. So you automatically find the white blood cell, because your dengue virus now already cannot infect cell anymore. It already be trapped, trapped. But you cannot neutralize it. The antibody cannot neutralize it. So what is happening? White blood cell go to eat them, because they want to remove the virus. The virus cannot in your blood, cannot in your body. They need to remove. So in theoretical they should remove, but now no. This dengue virus, when it enter into the cell, it will cause infection. So it will replicate using your host DNA, mechanism, transcription, translation to reproduce more and more dengue virus. The virus will replicate and package all these virus into, so it will produce more and more virus. So hence ADE, antigen dependent enhancement make the virus more easy enter to your cell. Compared to the primary one, primary is your virus need to find, dia kena cari punya. Tapi secondary punya dia tak perlu cari, just stay at there only because your antibody will find him. Your antibody find him, bind to that, but your antibody cannot neutralize the virus, so that your white blood cell will engulf the virus, and then this virus automatically enter into your white blood cell. White blood cell use all the host mechanism, transcription, translation process, packaging the virus, same like the HIV virus to produce more and more virus. So AD thus result in a greater burden of the infection, induce imbalance poor inflammatory and anti-inflammatory response. So it cause more severe situation. That's why it will cause dengue hemophagic fever. So this is more serious because dengue virus in secondary infection more easily enter to white blood cell compared to the first one, primary one. So this is a devil. So that's why we say that when the person get the second time dengue, it will get more serious, more severe condition compared to the first time. So also one of the reason that why dengue is very difficult to be treated. So, of course, you also learn about the symptom. So this is the symptom that can be found in the dengue virus. You can build yourself lah. So one of the major symptom is low platelet count lah. Low platelet count is one of the majority symptom. Also the statistic to discover you get the dengue. Also there are other type of the symptom you can build yourself. Okay, now we go through the prevention method. Okay, so prevention method, there are 3 prevention methods as stated by the MPM, social, economic, and biological. But for dengue, normally we're focusing on social and biological lah. So social, of course, is like us lah. Like us, what we can do. method is install mosquito net to your door, your window, so that your mosquito not easily enter into your house. And also remove any water from the container. Like the container, like the flower pot, tray, bucket, jar, all that. You need to remove all that to prevent the mosquito lay the egg on the water. And then another one is house to house away campaign. So how this campaign works? So normally like the volunteer or healthcare volunteer visit the household, educate the resident, educate the public so that how they can know about the knowledge about the dengue lah, how to prevent the uh transmission of the dengue caused by the mosquito and this mosquito. Also social media campaign, authorizing the social media, raise the awareness, discussion about the prevention method to enhance the public knowledge and also participation in the combating of the dengue lah. Okay. And also the this one is also one of the method. Wear light color normally is white color lah, and long sleeve, loosely fitting cloth and trousers so that your mosquito cannot bite you. Also waste management. Yeah, waste management also very important. Like normally water logging in your drain, longkang. Itu semua kena proper so that the mosquito will not lay the egg at there. Also the biological one. So biological there are two major method, there are five major method, 1, 2, 3, 4, 5. Well, first one is dengue vaccine. Yeah, for now, they are already have vaccine for dengue, but dengue for vaccine, but the effectiveness is not say that very good lah if compared to smallpox, because dengue have 4 virus. So usually you want to discover the dengue virus, dengue vaccine is very difficult. So but for now it's already got one that vaccine already have dengue to treat the dengue, which is the TAK 003 if not mistaken. TAK 003 that already approved by Malaysia. Already approved by Malaysia one. You go to clinic kesihatan or you go to any healthcare center, you want to dengue vaccine, already have. So normally this vaccine is actually live attenuated with dengue 2 virus, number two one. So it's more rapidly function if you receive if you get infected by the DNV2 lah, more effective. You get 1, 3, 4 not very effective lah. So this vaccine will trigger immune response as well lah. So all the work mechanism by the vaccine, you can just repeat what I am saying by saying previously in the vaccine topic. Also uses of the insecticide and plant derivative insecticide. So this one is to kill the Aedes mosquito. Aedes mosquito. So if you want to become more natural, you can use a plant derived insecticide instead of using chemical insecticide, which is more environmental friendly lah. So for example, you can use the plant from the pine, plant from the milk, plant from the aromatic gases that can already recognize can to kill the insect lah as well. So this is all about the first two method about the biological one. Next we go through another 3 we uses of the larvivorous fish and crustacean, meaning that apply the larvivorous fish that can eat the larva of the Aedes mosquito. So for example, fish like Porcelia viticula and also Mejocola for menses. So normally this advantage is cost effective lah. Cost effective, environmental friendly, and easily to be controlled. Another one is paratransgenesis. So normally this one is using the GMO technology, genetically modified technology, that using the GMO bacteria, genetic engineered bacteria that are we introduce the vector to colonize the vector population, hence limiting the transmission of the disease. Meaning that they introduce generate the bacteria, GMO bacteria that can introduce into the mosquito one. mosquito one. So that this mosquito, why this mosquito can limit the transmission of the dengue virus in the Aedes mosquito. Why? Because this bacteria provide the harmful effect to the Aedes mosquito. This regulate its sexual cycle, decrease the host competence, and interferes the developmental process of the vector species. So thereby it suppress the vector population. Meaning that this technique is that produce the GMO bacteria, and this bacteria actually is harmful to Aedes mosquito. So when they introduce this bacteria, infect this bacteria into the Aedes mosquito, the Aedes mosquito is dysregulate their sexual cycle, meaning that if they want to produce the next generation, reproduce the generation is very difficult. So that the point purpose is to reduce the population of the Aedes mosquito. So one of the example of the bacteria is using Wolbachia bacteria, and then another one is using of the sterile insect technique SIT. So normally this one is refer to the release of the laboratory sterilized male Aedes mosquito to the target population. So meaning that once this sterilized male mosquito is already released, this male mosquito can suppress the fecundity rate in female mosquito, reproduction rate lah for the female mosquito, consequently control the vector density in urban environment and transmission of vector-borne disease. So this mean that they change the uh release the sterilized male mosquito so that it can change the reproduction rate of the female mosquito to reduce the Aedes mosquito rate lah. So normally sterilized male were the potential competitor and can help the suppress the number of the offspring of the Aedes mosquito lah. Okay. So next we will go through the cholera, the second disease. So cholera for now is already rarely happening in Malaysia. Normally this disease is happening in the country which low developed country, poor country like Africa. Normally Africa country or in Malaysia also have. Normally is very poor lah. The family is very poor. Because this disease is transmitted through the contaminated water and food. If your water is clean, your food is clean, this disease will be directly cannot be transmitted. So this is a dehydrating diarrheal disease. Remain a major public health problem in low and middle income country, especially low income country lah, where your water sanitation and hygienic technique are inadequate. So the causes of this disease, your infection agent is by the bacteria we call as Vibrio cholera, like this. This is the structure. Structure also you need to remember because in exam they maybe just come out this kind of diagram, then you need to know this disease is called. So normally this bacteria, the major characteristic of this bacteria is they have flagellum. You can see there are the flagellum at there. So the scientific name is we call as Vibrio cholera. Okay. So in this disease, there are no vector. Vector is also Vibrio cholera because the vector is contaminated water. It is transmitted through the water only. This disease is stay at the water. Normally, it stay at the water and the food. So what is the sources of this bacteria? Normally, contaminated water and food. And food. So the transmission normally is between the individual with the same household in a high populated area in full infrastructure. So meaning that when you drink the water, drink this uncontaminated water, and then you stay at the household dekat rumah semua rapat-rapat punya, then share the spoon, share the fork, all that, or using the hand to eat, all that, you will easily to get the disease lah. Bacteria normally found in the slow flowing river, especially with a lot of backwater and coastal water. And some of the food also containing cholera. Some of the food like raw shellfish, raw oyster, if you not cook them, you also is the sources of the cholera as well. So normally this is the life cycle of the cholera. So normally when the cholera is normally stay at the water, water. When your water is already contaminated, you do not have any treatment. Although the cholera stay at there, and then your cholera will go at there, and when the human drink that water or food, it will pass from the polluted aquatic environment into the human gut, lah. So after that, you will get the incubation period. Normally is up to 5 days. Up to 5 days. When your cholera this bacteria starting to infect many cells in your body, half of your body, you will provide the symptom lah. The symptom that you will mention. The majority symptom is dehydration. So half of the severe cholera patient can die if proper treatment is not given. So your cholera cases can shed the bacteria from 2 days to 2 weeks of one set of infection. So this is a transmission of the cycle. So the major transmission is this. When you eat the contaminated water or food, you will get this. Okay. So normally in the cholera, this is the development of the cholera. When the viable cholera, this bacteria enter into your body, this will be happen. So for bacteria, normally your host cell in this by this bacteria is intestine cell, small intestine. No, is intestine. So it is intestine cell. So your bacteria will go to the intestine cell and release the enterotoxin. It will release the toxin. Release the toxin known as the collagen and the enzyme protease which act on the mucosal epithelial lining of the small intestine. So your collagen was starting to change the surface protein as well. So change the surface protein on the epithelial cell so that it become the receptor to the toxin collagen. So your collagen was starting to stimulate the epithelial cell to secrete large quantity of the chloride ion. Okay, so when the bacteria go and enter coming into the intestine cell like this, it was starting to secrete the enzyme we call as neuraminidase. In this diagram they don't show lah. It was starting to secrete the enzyme neuraminidase that will utilize the intestine epithelial sialic acid as a nutrient source to modify the intestine polycyclic ganglioside into GM1 receptor. So that's what I mentioned before. Meaning that bacteria will change the receptor on the small intestine, become GM1 receptor. The original receptor is recalled as a polycyanated ganglioside. But for now, due to the presence of the neuraminidase by this bacteria, it changes the receptor become GM1 receptor. So when this receptor changing, this bacteria at the same time will also produce the toxin, as I mentioned before. Toxin mentioned is collagen. It will secrete the toxin called collagen, and this collagen will bind to the GM1 receptor like this. So this blue color is GM1 receptor, and then you can see here this one is the toxin. This one is the toxin. So it bind to it, and this toxin will enter into the intestine cell by the endocytosis process. So the internalize of the toxin from the endosome will go to the Golgi apparatus and the endoplasmic reticulum to produce CTA1 protein here. So it will go bind into the Golgi apparatus or endoplasmic reticulum to produce the CTA1 protein. And this protein will activate the G protein. This protein will activate G protein in the intestine cell. So normal in the intestine cell. So when it activate the G protein, it will leading to the increase of the adenylyl cyclase activity, one of the enzyme. It will increase the adenylyl cyclase enzymatic activity, which cleave ATP to become CAMP. CAMP. And when you activate CAMP, meaning that it will activate protein kinase. So this is like the non-steroid hormone. You still remember non-steroid hormone, CNP, right? CNP, G protein, G protein, right? G protein activate adenylyl cyclase, adenylyl cyclase will cleave the ATP to become CMP, and then the activation of CMP will activate the protein kinase activity, like the non-steroid hormone mechanism one. Okay, after that, this protein kinase will cause will inhibit sodium chloride absorption. Meaning that sodium and chloride now cannot absorb anymore. Cannot be absorbed in the intestine. And phosphorylate the cystic fibrosis transmembrane conductor regulator (CFTR). So it will inhibit these two protein channel lah, I can say NHE and CFTR. NHE is sodium ion hydrogen ion exchanger, and then CFTR we call as the cyclic fibrosis transmembrane conductance regulator. More longer, and also there are also the chloride channel protein lah. Lead to efflux of the chloride ion into intestine lumen, induce a secretion of hydrogen carbonate ion, sodium ion, potassium ion, and water. Meaning that when you have protein kinase, your activity of the protein kinase activate, it inhibit NHE, meaning that sodium ion cannot be absorbed anymore. It cannot be absorbed by the intestinal cell. So if salt, you can imagine if the salt cannot be absorbed, meaning that water in the intestinal cell is higher now, high water potential. So water need to diffuse out from the cell. So it will lead to efflux of chloride ion. So your chloride ion inside the cell also need to efflux out, because sodium chloride, want to maintain the electrical balance, right? Because your sodium ion already cannot enter into the intestine cell, chloride ion cannot maintain in the cell one. They need to efflux out. Because they want to maintain the electrical nature. If they still inside the cell, meaning that this is already negatively charged, cannot. So chloride ion need to efflux out. So when your chloride ion efflux out, automatically your water and your bicarbonate ion will still also efflux out. So when your water water out from the cell, water potential of the cell was starting to follow loss of the chloride ion. Your water potential in the cell is higher, so your water potential still automatically out from the cell as well. So it is like a logic logic like the transport ion between the intestinal cell and the surrounding cell. I repeat again, when your protein kinase activity high and He is blocked, your sodium ion cannot absorb into the cell. Okay, so when you cannot absorb into the cell, chloride ion need to out from the cell. Out from the cell. When your chloride ion already lost from the cell, water potential inside the cell increase. So your water potential also out from the cell as well. Out from the cell. So doctor will diffuse out of the cell by osmosis. This will resulting in the violent inflammation of the intestine, and toxin prevent the normal absorption of the salt and water, resulting in vomit and severe dehydration. Meaning that there are many water outside of the cell, it will cause vomit and severe dehydration. So that's why we call main symptom of this disease is severe dehydration. You will vomit many water. Many water will go out from your body, because all of the water from your intestine cell is efflux out. And when there are more and more water, your intestine cell finally will die. It will die one, because you need to know cell also require water. If no water, then your cell also will die. So this is the development where what happens when your bacteria enter to the body. Okay, so this is the symptom. So the major symptom is severe dehydration, loss of the fluid leading to shock. So there are also other excessive stress, nausea, vomit, dry skin, leg cramp, dry mucus. So normally all these is like you already lose water. All when your water lose from your body, these are all the symptoms that you may have when you get infected by this bacteria. Okay, prevention. So this disease, want to prevent is very easy. You need to make a good hygienic practice. So first one, you need to ensure that your water is clean. Before you drink the water, you should boil the water. Boiling water is very important so that you can kill majority of the bacteria in the water. And then also for washing. Washing hand before food preparation, and also wash the food. Your hand before you do anything, especially you cooking, you should wash your hand. And also all the food, especially vegetable and fruits, you should wash properly, using properly. And of course, practicing good toilet hygienic practice by washing hand and clean water after visiting the toilet. And then of course you also need to need to wash your toilet frequently by using the detergent, wash down with the soapy water, detergent, and also properly wash your hand every time. Every time regular washing your hand with soap and clean water should be done before, during, and following food preparation, as well as when feeding children or yourself. So this is a good hygienic practice where this is a prevention. Okay. When you go through the biological way, so there are vaccine as well, cholera vaccine. So the example of the vaccine is the B subunit kill whole cell oral cholera vaccine. The name of the vaccine. So normally this vaccine is taken orally. This is orally, meaning that it does not require the just orally and direct stimulate the mucous immune system in the intestine. So it prevent your cholera, stimulating the intestinal and systemic immune defense against the viral cholera and its toxin, blocking the bacterial colonization, meaning that it block the bacteria, viral cholera, colonize, bind to the intestine cell, prevent it release. Yes. So normally vaccination in endemic areas should be contingent on the multi-skeletal intervention as part of the long-term plan for the cholera prevention and control. So that you need to this vaccine program, you need to work together with the government not only government as well, but the public society also need to work as well. Okay, so that's all about the cholera. And then the third one is TB, tuberculosis. The short form is TB. So tuberculosis, it is also one type of the airborne disease similar like COVID-19. But the infection agent is different. TB is caused by bacteria. It is caused by that bacteria, but COVID-19 is caused by virus. But the infection area is same. It also infect lung, and they are also airborne disease. They're transmitted by air. So the infection agent by this bacteria by the bacteria we call as Mycobacterium tuberculosis. Very easy. The genus name is Mycobacterium, and then the species name they just name it tuberculosis. Very easy. And there are also another one. This one is in male. In male one. Mycobacterium bovis. This one is found on raw cow milk. So that's why that the person we need to do a treatment for the cow milk. We cannot directly drink from the raw cow milk. You cannot directly drink the milk directly from the female cow. What? Excuse me. What? So it is a rod-shaped bacteria that will infect the macrophage at the alveolar cell in the lungs. So this is I already mentioned. The infection organ is lung, and then cell is macrophage at the alveolus. In the alveolus. So the transmission is airborne. So there are no vector in this disease. No vector, because the transmission method is airborne. So meaning that when you couching, talking, laughing, and you are very near to the infected person, you may easily to get the disease. So when you have the disease, aerosolization, meaning that you coughing, cough, sneezing, or talking very loud, all that you will release the air droplet, that containing TB, containing the bacteria. So that when the uninfected person exposed to this air droplet and inhale the air droplet, it will get the TB. It will infect your lung and you will get the TB. So normally the person with the active pulmonary TB will generate the droplet nuclear, which air droplet that containing the bacteria through the cough, singing, shouting, sneezing, or talking as well. Will spread the droplet into the atmosphere. So when you are close contact to this person, you will easily to be susceptible to the TB. So normally TB has been called plenty and quite justified a disease of accommodation, highlighting transmission of TB. TB is normally happening at a highly populated area. highly populated area because it's airborne disease. So normally, if you are very crowded, populated area, it is very easily to be spread. So similar like COVID-19. Totally similar like COVID-19. COVID-19. So normally in this disease normally this disease is coming in school, public transport, working place, healthcare facility, prison, penjara, and also mining area. Because all these areas are highly crowded area, high populated, highly crowded area. So yeah, this is a transmission. And also there are also another pathway by mycobacterial bovis, which through the cow. So when you drink the milk, which not pasteurized, the totally raw, pure milk from the cow, you don't cook any cooking, any treatment. When you directly drink it, you may get the TB. So this is by the cow. Okay, so the development. Okay, when you get the disease, your bacteria will starting go to your lung. When the alveolus lah, alveolus, and will go to the alveolus, will infect your macrophage in the alveolus. So this bacteria would be destroyed, and gut bacteria would be destroyed when the phagosome fused with a lysosome to form a phagolysosome where acidic enzyme kill pathogen. So this is normal situation. You need to know macrophage also leukocyte as well, white blood cell. So actually, it should kill the bacteria, but this bacteria will have the special mechanism that can block the maturation of the phagosome. So in macrophage, the phagosome cannot form. So due to no phagosome, so all this cannot form. No acidic enzyme. No, you cannot fuse with the lysosome. You cannot fuse the lysosome. So it cannot kill the bacteria. The bacteria stay at there. So it resists the degradation by reactive oxygen, oxygen and nitrogen species. Because of the bacteria thick waxy microlith cell wall. So this bacteria, their cell wall is spatial, is very thick that can be resist the degradation. So that the bacteria already infect the macrophage. So it will reproduce inside the macrophage. Why? Because it already delay the adaptive immune response. You need to know macrophage act as antigen presenting cell. So it should display the antigen fragment of this bacteria. But for now, due to already cannot digest this bacteria, the bacteria stay inside the cell, stay inside the macrophage, it already delay. So when the macrophage display the antigen, it already very long time already. Long time already. So during this period, your bacteria still can replicate inside the macrophage. So it replicate inside macrophage, release more and more bacteria and infect other type of the macrophage. So the macrophage will not die. It still maybe can display the antigen, but when the macrophage display the antigen to trigger the adaptive immune response, it already too late. So it already cause all these symptoms. So normally causing, wheezing, fever, all these are the basic symptoms for this disease. So there are also other lah. You can read yourself. So for prevention, okay. So one of the method recognizing by WHO is using FAST method, which find cases actively A, separate safely is S, and treat D effectively based on rapid drug susceptibility testing. So this is the method. So how this method can prevent? So you need to find the case actively. When you get the suspect case, you need to find the case actively. So you need to know that TB is easily to be spreaded by the institution by coughing or sneezing with unsuspected TB or unsuspected drug resistance. So most potentially, the infectious patient can be identified very easily. So when you see the person which already have cough or sneezing, you need to do a very rapidly test, the screening diagnosing to diagnose this person as the TB. So this will reduce the duration of the institutional exposure through effective treatment. Transmission among patient and to healthcare worker will be reduced proportionally. So this method actually is to based on the assumption that most transmission occur not from non-TB patient, but also from the person with unsuspected TB. So you already know that, oh, you got TB already. So the scientific side, the government should check the cases based on that person surrounding, so that they can quickly isolate all the susceptible cases to prevent the disease spread more serious to the environment, to the population, sorry. So population. However, this method normally is require government lah. Normal person cannot do lah. It require government. Okay. Government. Okay. Another method is directly observed therapy, DOT, also by the World Health Organization. So normally this one is to train the care worker, the doctor in the healthcare worker in the hospital one, with the prescribed TB drug, and watch the patient swallow every dose to ensure the patient take medication under supervision for 6 to 8 months. 6 to 8 months. Meaning that when the person get the TB, you need to ensure that this patient get the drug under the supervision up to 8 months. So it cannot, because we cannot predict that who take the medication as director and who will not. Who will not. Because we don't know when the patient go out from the hospital, you take the drug or not. So you need to have the worker that already trained to make sure that patient take the drug, take the medication every time. So it help the TB patient be treated more quickly, prevent the TB from spreading to each other, decreasing the risk of drug resistance, and also decrease the chance of the treatment of failure and relapse. Because you need to know TB is caused by bacteria. So in bacteria, normally you can use antibiotic to kill it. So if you want to kill the bacteria completely, you need to use antibiotic continuously. You cannot stop the antibiotic. If you stop the antibiotic, you will get the drug-resistant bacteria. Meaning that bacteria already resist to that drug. If you want to kill this bacteria again using the same drug, already cannot. Because it already can survive already. So that's why you need to have supervision. Ensure that the patient eat the drug continuously until end. So this is what we say as directly observed therapy. And also transmission control, not only through the breathing share air with an infectious disease, but also need to separate all the suspected infectious individuals to prevent the transmission. So that's all I already mentioned. Also early detection. You need to active case finding, like the active case finding, diagnose it very fast to diagnose. When you get the TB patient, you need to active find the case, diagnose the case, and also the government also play the role in prevent the TB as well. So the biological, so there are also the vaccine we call as BCG vaccine. Vaccine that the bacteria is using is Mycobacterium bovis from a cow one. Not from a bacteria they use from the cow one that can activate the immune system. So normally this vaccine is recommended to the children, to the children. However, normally this vaccine only protect against childhood form of the TB and do not provide the protection for the adult one. So this vaccination is not suitable for the malnourished children as their diet do not have enough protein to make antibodies. So normally this vaccine is more suitable for children. Adult actually not very. You still can do it, but the effectiveness is not very high. For now, in the case, in the treatment of the TB vaccine, there are so far no other vaccine yet in the world to introduce using the Mycobacterium tuberculosis. Okay, the fourth one, which malaria. Okay, so malaria actually similar like dengue because the transmission vector is by female Anopheles mosquito, but the infection agent is different. Dengue is using virus, but malaria is using protozoa. The infection agent is protozoa. So there are many scientific names here. Plasmodium, falciparum, vivax, malaria, OV, and Knowlesi. You just memorize one is already enough lah. And then the vector is female infected Anopheles mosquito. So the mosquito, the genus name is Anopheles. So yeah. So okay. So the transmission is similar like dengue. We using the mosquito, Anopheles mosquito, but the development is different. So this is a cycle of the plasmodium cycle. Like okay, first of all, when the mosquito is already infected with the plasmodium, anyone plasmodium, how it infected? It bite the blood from the infected person. When it bite the blood, you can see here. When it bite the infected person, it will inject the blood. Take the blood from the infected person into their gut. So the sporozoid, the terms we call a sporozoid will enter the protozoa, injected into a host bloodstream by the mosquito saliva. So inside the female mosquito now, they containing sporozoid. So this sporozoid will be injected into the hostile, into the human blood lah. Human blood lah. So the sporozoid will enter into the bloodstream and go to the liver. So now it is staged in the liver. It will go to the liver first. So it will infect the liver cell, which is hepatocytes. It will infect the hepatocyte and multiply into the merozoid. It will multiply at there and release to form merozoid. Now it's not called sporozoid, it becomes merozoid. Okay. Now this merozoid was starting to infect the red blood cell. This merozoid will go to the bloodstream and then it will infect the red blood cell, erythrocyte. So they will develop into the ring form called tropozoid, like this, tropozoid, and produce further merozoid, meaning that this merozoid will be reproduced in red blood cell. It will produce more and more merozoid. So when they produce more and more merozoid, the red blood cell will be burst. This will cause burst of the red blood cell and burst of a red blood cell and release many merozoid, and this merozoid will infect other red blood cell again. So as a result of this extensive division, millions of merozoid will be present in the human blood. Okay, so what happened next is that your merozoid, your small proportion of the blood stage parasite will develop into the sexual stage called gametocyte. That reach the dermis and they were taken up by another mosquito. Meaning that some of the merozoid will develop into the gametocyte. They will develop into the gametocyte. They will not infect the red blood cell anymore, develop into the gametocyte. So this gametocyte will be taken by the Anopheles mosquito. So there will another Anopheles mosquito go to bite this infected person, and it will take out the merozoid, the gametocyte. Take out the gametocyte. So after that, this gametocyte will enter into the Anopheles mosquito body lah. So it will undergo 5 distinct morphological stage during development. So it will develop because it is still gamete. It is gamete. So it will develop in the female in the mosquito body. So on injection, the mosquito midgut major gametophyte will aggress from their host cell, differentiate into male and female gamete. So in the body, it will develop into male and female gamete, and fertilization happen to form zygote. Like this. So once the zygote form, it will undergo meiosis to form sporozoid. It will form sporozoid that will reach the salivary gland, and now it will begin. It will be ready to infect another person. So when this mosquito go to bite another uninfected person, it will release the sporozoid into there. So this is a cycle. So firstly, sporozoid You just say spores in the salivary gland enter into the body and then go to the bloodstream, go to the liver, and then infect the liver to produce merozoite. So this merozoite will infect the red blood cell and then the red blood cell burst, release more and more merozoite. And some of the merozoite will develop into gametocyte. And this gametocyte will be taken out by another mosquito. And then after that, it will develop into male and female gamete in the mosquito body, mosquito meat gut. And then to form a zygote, and this zygote will undergo meiosis to form sporozoite. Sporozoite. And then sporozoite will go to the salivary gland and prepare to transmit it to another uninfected person by biting them. So that's all about the transmission and development of the malaria. Yeah. Okay, so this is a symptom of the malaria. You can read yourself lah. So this is a symptom. Okay, so the prevention method for social are actually similar like dengue. You can copy and paste from the dengue one. So I think I don't need to explain much. Mosquito name, remove any worker, blah blah blah. I just want to explain this. Chemopocylacix. Chemopocylacix. So normally this one is the one type of the drug. One of the drug we call as chemopocylacix is administered as a drug active against the pre-erythrocytic malaria parasite. So meaning that this drug can suppress the sporozoite infect the blood cell. So no blood cell. So that the cycle will stop here. Meaning that they produce the merozoite, but this merozoite cannot infect the red blood cell anymore. It suppress the formation of many merozoite lah. Merozoite. So normally this drug must taken at least 4 weeks lah. 4 weeks. Yeah. And then another one is here. So another one is the biological lah. So biological also similar like dengue. You also can use the lativorous fish that can eat larva, and also larva. So but for now I will need to focus on here lah. Insecticide treated nets. You can use the nets that already treated with insecticide. So to provide further protection by killing the insect coming in contact with the net. Also, you can indoor residual spraying. You spray the insecticide in the wall and floor of a house. So normally you need to recommend to use a natural one environmental friendly one Okay. Another one is attractive toxic sugar bite. Okay, so this one is use of the fruit or flower that can be act as the attractant, act as a feeding stimulant and oral toxin to kill the mosquito. So this actually is to one of the natural insecticide to kill the mosquito lah. This intervention is technologically and operational simple, environment friendly, cheap, and suitable for controlling malaria in low middle income country. And also habitat modification. You need to change the land and water, including landscaping, drainage of surface water, land reclamation and filling, to be removed the area using the simple materials. So that the mosquito not easily to lay the egg inside there. Okay, so yeah, that's all about the malaria. Next, we go through the last disease, which is the hand, foot, and mouth disease. Also, this is the new syllabus in the disease. This is, I think, this one, sorry, because I copy. This one, you know this, infection of blood, this one is for malaria, but you know this. So actually, this disease is caused by virus. This disease is caused by virus called enterovirus. And then the species one, there are two major types of species. It is enterovirus 71, EV71, and another one is Coxsackie virus. But you just know enterovirus 71 is already enough lah. EV71. So this is a symptom lah. You can see when the person who get this kind of disease, your mouth, your hand, your foot will have that spot. So that's why it say hand, foot, and mouth disease. It will infect your hand, foot, and mouth. You will get the red spot, and bottom of the feet is painful spot in the mouth and the throat. Okay. So the transmission. Okay. So normally this disease is transmitted by direct contact with nose, thorax discharge, saliva, fluid, and from blister or the stool of the infected person. Meaning that it's directly infected from person to person by direct contact. Normally it's through the nose or saliva. So that's why that this disease is commonly found in baby sister kindergarten. Especially in kindergarten, because baby they do not have the knowledge, right? They do not have the knowledge of the about the hygienic practice. Normally they play using the saliva, right? Not using saliva. They do not clean their hand. They just anything they just eat, right? Because maybe they do not have the knowledge. They cannot modify the information yet. Anything they see, they just eat, right? They just eat and play, and then play, and then they share it to their friends, and their friends touch it. Then the baby always like to use put their hand into their mouth, right? And then they put into their mouth, then they get this kind of disease. So this disease also can be transmitted by fecal oral route by contact with a contaminated object. Meaning that fecal oral route, meaning that this is also by baby lah. Meaning that when the baby tahi, mereka berat kan, berat mereka tak tahu punya. Maybe mereka akan makan tahi tu, baby punya, baby. Or contact with a contaminated object. Maybe mereka berat dekat toys dia, dekat permainan dia. Mereka pun tak tahu, mereka just play je. Kalau cikgu dia tak nampak, mereka main je. They contact with a contaminated object lah. So akan get this disease lah. Also, lack of access to clean water, poor personal hygiene, overcrowding, large family size. So all this is daycare center. So incubation rate is estimated in about 3 to 6 days, and children are highly susceptible because of immature immune system and clustering and pre-kindergarten stage. So all this. Alright, this is about the development. So all this is more complicated one. I just try to explain more easy lah. So this virus, the EV71 virus, they will bind to the virus receptor on the surface of the epithelial cell. Normally the target cell of this virus is intestine epithelial cell or funginea epithelial cell. Funginea epithelial cell. So they will replicate at there. So it will replicate in the submucosal lymphoid tissue of the lower intestine to a lesser extent the fungus. So after that, the virus will replicate at there. Endocytosis, you will go through endocytosis. Is triggered to release the RNA virus, to produce more and more virus. So it will use all the host mechanism, transcription, translation process, and release more virus. So this virus not only infect the intestine cell, at the end it will also infect many organ. When they enter into the bloodstream, they will also infect your brain, spinal cord, your brain, your heart, and even your lung as well. But the primary site of the infection is epithelial cell, intestine epithelial cell. So what is the effect is the replication of the virus at this site and the associated inflammatory response are responsible for the clinical manifestation and complication of the disease. So you will provide many serious inflammation. So these are all the symptoms. So one of the major symptoms is this red spot appear a few days later on the hand and the bottom of the feet. This is a major symptom. Also painful spot. Here. Okay. So we want to prevent this disease. You should provide good personal hygienic practice. So in the daily kindergarten, you should educate especially daily care, not baby, 5 years old, 6 years old, that at that age, you should educate them to do the good hand washing, how to do a proper hand washing after toilet, diaper change, and before meal as well. And also avoid the close contact with infected individual, and sharing of personal item are protective factor and should be encouraged. Okay, and also pregnant, for those who are pregnant, women also should avoid contact with the individual with this disease. In this disease, if you contact it, your baby, your fetus maybe will have infected. Also, also prevent, avoid bring your baby to the public area lah. Area lah, like cinema, shopping center, all that. If you bring them to that, they are easily to susceptible to that disease. And clean all the toys, clean and sanitize all the toys or the table before use and after use. Avoid touching your face using with unwashed hand, especially your eyes, nose, and mouth. So this is all you need to educate the children. And also another one is the management outbreak in kindergarten or primary school in Malaysia lah. This one is based on the MOH, Ministry of Health, MOH. So if normally in Malaysia SOP, if there are the cases reported in kindergarten or primary school, the case detection, active case detection should be carried out among the contact in the institution and family, meaning that the whole family, the whole school, uh, school need to be contact. They need to contact every children in that school, in that school, in the school. So health education and supervisor of the institute of the disease, mode of transmission, importance of good hygienic is should be carried out. So the government also need to responsible to provide the good hygienic practice to the school teacher. They need to educate them. So, and also all the teacher and the staff in the school who be alerted to look out for the children with fever, rash, litter, or palm and sores to isolate them immediately. So if you see the cases, the school need the alert. They need to isolate them and directly call the parents. Let them know you already get this disease. They need to get the immediately treatment everywhere and need to close the school for two incubation period, which is normally 10 days, 10 days from the date of onset of the last case. Hence, and also need to do the sanitization to the whole school. Okay. This is a good hygiene practice. You should educate them how they want how educate the children. Okay. So about the prevention in terms of economy. Okay. So the first one is improve clinical case management service, particularly for severe manifestation requiring intensive medical care. So this one is to ensure that everyone in the healthcare staff and nurse and doctor is aware with this disease. So that they already have information about this disease, so that they can provide the provide the quickly treatment to the patient. uh, to the patient. And then second one is establish and strengthen the surveillance. So they need to functionize the surveillance system to provide the timely data and information for risk assessment and subsequent decision making about appropriate public health intervention. So they need to monitor and evaluate the impact of such intervention. So you need to always monitor the surveillance patient. Okay. Next is coordinated risk communication, meaning that information resulting from the clinical laboratory investigation must be directly go pass through the exposed to the public. You cannot store the information. The government should not menyembunyikan isu ini. Sapu dekat dalam, tak boleh. You should public out. You should announce what is happening now. Provide the accurate, timely, and consistent information. So that everyone know. They can know what is happening. What is happening. And then the final one is providing assistance to kindergarten, daycare facility, and school during outbreak. So you need to government or other NGO should provide lah guidance, how to the teacher, how to how to overcome it, how to when you face the HFMD disease, how do you want to uh what are the protocol that you need to perform to control, to handle this disease. So yeah, that's all. And then the final one is the biological pathway. But so far in the world for now, there are vaccine. They already discovered the vaccine, but in Malaysia still not be approved yet. So they already got discovered EV71 and CVX16 vaccine, but in Malaysia they are still not be recognized by the government yet lah. Our government still not recognize this vaccine yet. So, but in the real, they already published this vaccine, already approved by FDA. FDA, yeah. So normally this vaccine is containing formerly inactivated EV71 vaccine with an alarm adjuvant to boost the immune response. Also for CVA16 is live attenuated vaccine normally is to stimulate the secretory IgA at the mucous region. For EV71 is to stimulate systemic IgG antibodies. So yeah, that's all about the hand, foot, and mouth disease, HFND. Okay. So that's all about this chapter, and also that's all about this semester as well. So thanks for those who are watching from the beginning until end, and also from the first chapter until this chapter. So hopefully this video can provide the clear explanation to you guys what you will learn in this chapter. So if you have any question, you just type in the chat box or comment area, or you can join my Telegram group as well. So I think that's all about this video. So hopefully you guys can score well in this semester and do not take the repeat. So thanks for your supporting and goodbye.